So tetrad analysis with UnlinkedGenes, measuring distance to the centromere. Right. Now, suppose we're not going to work with Lu and arg and met. We're just going to talk about big A and little a and big B and little b. Suppose you have these two haploid strains, big A, big B, little a, little b, right? And these genes are unlinked. So you would maybe say, for example, if you mated them and you starved them, grew them up, starved them, made them do meiosis, you'd have maybe a configuration that looked like this in meiosis 1 after S phase. Here's the chromosome that A resides on, and then here's the chromosome that B resides on. Okay. Now let's suppose, just for the purposes of explanation, that A is really closely linked to the centromere. Right? So big A there, and little a there. But B not so much. Okay, so maybe B is out here, and then out in this yonder region. Again, I sort of show the parental genotypes on top and and on the bottom as groups. Now, just to remind you that the parental ditypes are equally as frequent as the non-parental ditypes in this scenario, because in one pattern of segregation, big A and big B stay together and little a and little b stay together, but it's equally likely that this could go this way and this could go this way. In that particular case, you'd be producing big A, little b spores, or little a, big B spores. Again, the parental genotypes, the parental ditypes and non-parental ditypes basically equal. All right. Now, what happens if there is crossing over between a marker and the centromere? Okay, well here we said that they're too close. So let's suppose we have a crossover here, right? What's worth thinking about, what type of tetrad would this produce? Right? Well, let's just think about it. And we're not going to go through all the sort of gymnastics of meiosis right now, except to sort of say that the way I like to think about this is I'd like to think about each of the sort of haploid products segregating and how that would occur in this particular scenario. So here what we would basically be demonstrating is big A would be going with big B, right? So if we have our tetrad here, it would produce a spore of that genotype, right? And then the next spore would be big A as well, but notice something. Now it's going to be, this centromere now loses its linkage to big B. It now carries a little b, so it's going to drag a little b up with it, and you'll have a sister chromatid that is recombinant essentially. And so that would produce big A, little b spore, all right? And then this one down all the way at the bottom would produce a parental genotype, all right? Little a, little b. But now this little a here on this side, right, we're going to say segregates with that big B. So there's 4 different types of spores here, and so we call this a tetratype, right? And the degree to which this happens is a function of how far one of those genes is away from the centromere. And it turns out you can actually measure the genetic distance with this little formula, all right? So if you know, if both of them, it's a little tricky. If both of them are unlinked, it's a little messy, right? Because you can kind of, well, which way goes which with the single crossover. But if you assume one of them is linked and the other one is, one is like very closely linked to the centromere, and the other one has some genetic distance that you're trying to measure, all right? The genetic distance between the gene of interest, in this particular case B, and the centromere is given by this little formula, right? It's just simply the number of tetratypes that you observe, all right, times a half. And the reason it's times a half is, of those tetratypes, right, only half of the meiotic products, only the half of the haploid products that come out of that particular meiosis will in fact be recombinant with respect to the centromere. The other half are the parentals, right? The one on the bottom and the one on the top. It's only those sort of 2 here that contribute to the crossing over that is estimated in looking at genotypes. And so it's 1/2 times the number of total tetratypes times the total number of tetrads that you've analyzed, and that will give, you know, a frequency, and then you would multiply by 100 to give you the centromere distance in this particular case between B and the centromere.